to migrate at different times. So again, these are largely correlative studies. So first of all, just trying to establish a correlation between a specific region of DNA and our phenotype of interest. Getting from that to actually a functional question is a lot more work. Okay, so let's start off. So what is this field we call population genetics? So really, population genetics in and of itself is really nothing more than a set of statistical models for interpreting genotypes. It's really just a subdiscipline of statistics. So essentially what we do is we collect, genetic or genotypic data on our animals or plants, whatever of interest, and then use a series of statistical models, collectively known as population genetics, to essentially look at the questions of, do particular genetic variants that we see, will they become more or less common over time, and why? And then also estimated the effects of population structure and different evolutionary processes on allelic and genotypic frequencies. So we look at how does genetic structure change over time. Oops, I'm sorry, went a little bit too quick. So generally the models that we use look at, how does genetic structure change over time. So the models we use actually use deviations from some fundamental assumptions to detect changes in trends. So such things as mutation, migration, do animals or fish or plants, whatever, do they move, do they disperse? When they disperse, what effect does that have on their underlying genetics? What genes are they taking with them? Also the effects of natural selection, which is an adaptive process, to where, different environmental cues, in the world of animal plant breeding, selection is artificial, and that you're intentionally making different traits change. So that's an adaptive process, as opposed to what we call genetic drift, which is more of a stochastic process, in which our genetics are actually changing because of basically random sampling from one generation to the next. So in that case, the difference in genetics that we see is the effect of purely random and stochastic events, as opposed to something that is adaptive in nature. And then also non-random mating. So particularly if you're looking at, say like large mammals, for example, with a very strong herd structure, obviously you might have, maybe like one dominant buck or something who, gets, kind of the bulk of the matings. Is there a certain selection going on to where, individual animals, get to reproduce based on some physical characteristic? So another thing I want to point out too is many of the population genetic models that we use were actually derived in the 1930s by Wright and Fisher. And it was interesting, a lot of the models that we used said, they're developed not quite 100 years ago now, but a lot of them were developed just from physical observations of phenotypes and how those change through breeding programs. So a lot of it was developed, before we even knew what A's, T's, G's, and C's were. But surprisingly, a lot of those models still hold up well. Obviously, there's been changes to them over time, but a lot of the fundamental ideas that were developed in the 1930s and 1940s are still correct and still hold. So there's one particular thing that I do want to spend a couple minutes talking about because it's worthwhile. This concept of effective population size. This is probably the one single population genetic parameter that I hear people talk about the most. And a lot of times, folks will talk about it, they'll mention it. I often hear it, even geneticists often, sometimes you may be using it more incorrectly than correctly. So this idea of an effective population size is very critical when you're looking at conservation, and recovery questions. The reason is because you can go out, you can do a physical survey, like a physical census of your target organism and say, okay, I surveyed this population. I think I have 500, breeding adults. Okay, I'm good to go. So I'm not worried. But effective size is a parameter that we estimate that actually is actually used to determine what the true inbreeding rate is, and also how big of a role stochastic processes like genetic drift are going to play in how that population performs. Now, a lot of times when you maybe go out and I survey and say, okay, I think I have 500, adults here, So I think I'm pretty good. I don't have to worry about inbreeding. But effective size is how that population is truly behaving in a genetic sense. And it's not uncommon for the effective size to actually be maybe 10% of your population census size. And again, it's a critical metric that we use because it directly informs what the rate of inbreeding is going to be. It directly informs how significant genetic drift is going to be. And genetic drift and adaptive processes kind of fight one another in that you want probably ability to adapt to new conditions to drive the bus as far as being resilient over time. But genetic drift kind of works against you in that you lose genetic variability because of random processes. So again, I just want to mention that a bit. Said effective size is a metric you might hear people talk about, but it's often poorly understood, but a critically important one that we deal with quite a bit. So just again, generally some conservation and management questions that we're involved in is pretty much, again, the larger question of how is the genetic diversity that we see actually spread across the landscape? Because this really informs different restoration, management, recovery questions in terms of identifying where are actual true population units. For instance, you might survey an area and find, okay, I have a lot of individuals living here, but maybe they're actually not all reproducing with one another. Maybe I have some individuals that are resident and others that are migratory. So even though there's a lot of them here, maybe I actually have a pretty highly fractured population structure, meaning that my reproductive units are smaller than I think, which increases our rate of risk for inbreeding, increases our risk for genetic drift and non-adaptive processes to happen. Patterns of connectivity is a bigger and bigger issue. Obviously, as we all know, that habitat fragmentation and wildlife corridors is a very big issue, for us going forward. When you look at patterns of connectivity, some of the things that we are able to gather from our genetic information and estimate, you look at things like actual rates of migration. Also, is there sex bias dispersal? Do the males of one species tend to disperse and reproduce a lot farther than the females do? Are we actually seeing more and more differentiation amongst our different populations, meaning that maybe ones that used to exchange individuals can't anymore? Hybridization is another very significant issue that we directly inform. That just to use a trout for an example, here in California, and I'm going to talk about this in a bit. California is a kind of a hotbed for diversity of Olmicus diversity. So Olmicus is the rainbow trout. It also encompasses steelhead, our golden trout, red bands. So there's been pretty significant issues in the past and currently with identifying, okay, which population units are pure, which populations are actually hybrids, either with a different species or a different subspecies or perhaps a different strain of trout. And that directly informs which populations might be candidates for listing, which populations, if we have to go in and do rescues, which ones do we want to save, which ones can we let go. Also, if a certain population blinks out because of, say, like fire, drought, something like that, where else are we going to go? What population will be the appropriate one to go and select to recolonize that habitat with, maybe once after restoration has been completed. Adaptive variation is something directly related from the genomics area to where we now have the ability to maybe find genetic markers that might have a significant correlation with a specific phenotype of interest. I used migration timing in Chinook salmon as an example. Chinook salmon, as folks know, basically any salmonid, probably have maybe, I don't want to say a disproportionate amount of resources poured at them, but population genetics, especially on the fishery side, all originated mainly with work on Pacific salmon. There's a lot of money and a lot of work being done on trying to find specific regions of the genome and specific markers that are associated with, say, like migration timing in salmonids. So all the work that we do, the metrics that we find, the patterns that we're trying to identify, really feed into the larger question of what do we prioritize for, say, restoration, recovery, and management. So we look at various things like our population structure. which populations are at the highest risk for inbreeding. Do we actually need to do managed translocations to try to stem the risk of inbreeding? And also, if a population becomes locally extirpated, and then we feel comfortable kind of repopulating it, where do we go? What populations do we use? What's the appropriate stock to use, or strain, or whatever, to reintroduce animals, fish, plants, what have you, to that new habitat? What is most closely mirrors, what may have been there before they were extirpated? So generally, what we aim to do on a conservation, genetics, plus biodiversity world is to try to minimize our loss of genetic diversity overall. So we know that generally a species needs diversity to adapt to new conditions. So that's what goes into establishing resiliency. You might hear folks refer to this as what we call the portfolio effect, to where maybe you have like the species as a whole statewide, but you have all these other little, perhaps independent or semi-independent population units. Each of those has, kind of its own, say, collection of genetics, for lack of a various phrase. But when you look at that portfolio across the species as a whole, that's what we want to maintain. Generally, you just want to minimize the loss of diversity on the species level, as the species, needs new tools in the toolbox to adapt to new conditions. We all know probably examples where you might have a particular species or subspecies or population that kind of lives in one very distinct location. It's been there a long time, has very, very little genetic diversity. It's probably because it's become very well adapted to that very specific environment that it lives in now. Thing is, it's done well, it's persisted, it lives there now. But then the question becomes, in the future, if the environment changes, in those individuals that have very little diversity, now, even though they're very well adapted to the conditions as they exist now, should those conditions change, they may not have the tools they need to be able to adapt. But the question of, well, when do you step in? When do you actually try to introduce something to increase diversity? That kind of goes to the genetic rescue question. That's a very complex discussion to have, and very, very species specific, very situationally dependent. So the next point on my slide about using adaptive variation for management decisions is pretty hotly debated and pretty risky, especially on a genetic, if you're using genetic markers to inform these types of decisions. Reason is, as I mentioned in a few slides back, is in the genomics era, a lot of what is done and the approach taken to identify specific, say, stretches of A's, T's, G's, and C's that are associated with a phenotype of interest. Keep in mind, many of those are correlative studies. So we've correlated maybe specific regions or specific genetic regions with a trait of interest. But in almost every case, maybe not almost every case, but I say in certainly the majority of cases, again, they're correlative studies, not causative studies, in the sense that we haven't really zeroed in on the exact gene or genetic sequence that is truly affecting the physiology of the animal and how that happens. So an important point to be taken from that is that sometimes these correlations, they might be specific to the actual study population of interest. And that taking that information from one population and extrapolating it to a different one, or even perhaps maybe a different subspecies, is pretty risky because that correlation may not hold. So it may or may not hold. So again, just be really careful about trying to use that type of what I call AGV or adaptive genetic variation when you're doing conservation and management decisions. Again, that's very kind of a situationally dependent type of thing. So if you're in that situation, certainly reach out to the lab, and we'll be more than happy to discuss thoughts with that. And the thought on how to use that in a conservation setting said it's being pretty hotly debated. It's a type of thing where the science is way ahead of ESA and CISA. So it's definitely a work in progress. So they talk about AGV or adaptive genetic variation in conservation. Again, there's a lot of papers out there. They're coming out almost daily. There was one that came out a couple of years ago that I put on here by Waples and Lindley. They looked at the genetic basis of adult migration timing in Pacific salmonids. Definitely a worthwhile paper to read if you're interested in this type of thing, mainly because they look at the question of how, when, where is it appropriate to use sort of genomic and adaptive information and genetic markers when you're looking at defining conservation units, you know, in the eyes of ESA. Again, it's a very dynamic area of discussion right now. Okay, so I'm going to shift gears here a little bit. Want to keep moving along here. So how do we generally do workflow in the lab? So obviously our genetics project starts with field collection of tissue samples. At least in the fish world, those then go to our Central Valley Tissue Archive in Sacramento, where they are properly archived and then split to be sent out either to people we're collaborating with or for us. We then do DNA extraction. So starting with step 3 is done actually at the genetics lab itself for projects that we're doing. So we do DNA extraction, then genotyping, meaning the collection of DNA information, followed by the data analyses, and then the reporting steps. I always get the question, well, how long does it take to do this? Well, my answer is it depends. Sometimes these asks for us are a very straightforward process, and we can turn them around fairly quickly. Other times, if it's a more larger project level type of ask, it might be, you know, several months to a year or so. Again, it all depends. I'm just going to, so next slide here, just really quickly, want to show you, I just like showing these off. They're pretty cool. This is what we use for genotyping SNPs, although we are currently slowly in the process of transitioning to genotyping by sequencing. But these are a microfluidic device we use in our laboratory for collecting genetic information on a lot of our projects. So it's a single-use piece of plastic that's about $600 a pop. But on one side, we can load 96 DNA samples. On the other side, we load 96 different genotyping assays, meaning we type 96 different genetic markers. And then using what's called microfluidics, we load this piece of plastic, put it in one of our instruments, and it does basically a full factorial combination. So it's essentially doing about 9,200 individual picoliter-sized PCR reactions in the center and giving us back data. So it's about 4 hours to set one of these up and run it, but from that we basically get about 9,200 data points. So we could usually run a couple of these in a day in the lab. So I think about just the cost. We basically go through about the monthly cost of a scientific aid salary in about a day and a half when we run these things. All right, so I'm going to give you some examples of more practical examples of how we've been directly interacting with agency biologists on some of our projects. So recently completed one was a SWIG-funded study on the Eagle Lake rainbow trout. So Eagle Lake is over on the east side of the Sierras, home to a native subspecies of rainbow trout, obviously called the Eagle Lake rainbow. It's a kind of a high altitude, high alkaline lake, very different water chemistry. It's also home to basically a Lahontan native fish assemblage. So kind of a very unique environment. Again, kind of on the east side of the Sierras, probably a remnant of the old Lake Lahontan at one time. So there's a SWIG-funded study. The Eagle Lake rainbow trout has been previously petitioned for listing. Listings were found to be not warranted because we run actually a hatchery operation. So all the spawning, all the reproduction that's done in Eagle Lake is done basically by us, by the department. Their one main water source is Pine Creek, and that's blocked by a weir, where we collect fish and do all the spawning. Eggs are taken to Dara and Crystal Lake hatcheries, where they're then grown out, and then the lake is repopulated. So one of the questions was, well, how good of a job are we doing at preserving diversity in these fish? Since all the reproduction that's done is essentially done by us. So we found, fortunately, Region 1 actually had some old fish scales collected from the early 70s from Eagle Lake, so we were able to analyze those as well, and then look at maybe long-term trends. So our deliverable from this was recommendations for the genetic management of Eagle Lake rainbow trout. And so I know some of the writing on here on the PCA plot's kind of hard to see. We have, so let's people's slides catch up a little bit. So I'm going to try to use my mouse. Again, this is kind of my first time presenting this way too, so bear with me. So kind of this, I don't know if you can see my mouse or not, but kind of the big sort of reddish colored, say, shotgun blast of data points to the right side of our PCA plot is essentially all of our Eagle Lake samples going back to the early 70s. So what we find from this is that Eagle Lake rainbow is pretty unique, but it's also been fairly stable genetically since the 1970s. Even in spite of having basically all the reproduction being done, the collection of broodstock spawning and reintroduction into the lake. Interestingly, just below our cluster of Eagle Lake samples, kind of the yellowish cluster just below, were individuals we collected from Pine Creek itself. So Pine Creek is one of the main water sources for Eagle Lake. It was also the primary historical spawning ground for the Eagle Lake rainbow since they're strongly adfluvial. So in the past handful of years, we've started letting fish selectively pass the weir and go upstream to reproduce. So we know quite a few adult fish were passed the weir, and then the field crews that recovered actually juvenile Eagle Lake rainbows in Pine Creek. So the question was, well, there's reproduction going on, but how many fish was it? So it's kind of a really big question in that we know we passed, I don't remember what the number was, say we passed 100 adults past the weir, and then we found juveniles. So an area where we really came into play on this on the genetic side was that we could look at these collections of juveniles and estimate basically relative spawner success. So what we found was that I think we found that probably about maybe 10 to 20 actual individuals reproduce successfully. So again, just another way that we help on more of a practical sense, and that we can look at relative spawner success as well. So another one, I'm going to leave this to Kristen to talk about more in her upcoming slide on eDNA. So this was kind of a big project that we did, kind of our first big in-house eDNA survey in Silver King Creek. Again, this was primarily Kristen's project she was working on, but I just mention it here real briefly because it was kind of our first big lift that we actually did basically all the lab work ourselves within the department, kind of very first time for eDNA. So I'm going to leave that more for Kristen to talk to folks about in a little bit. So this is another ongoing project that we have. It's basically more of a service-oriented project, but we do Chinook salmon genetic stock ID from adult Chinook salmon that are actually captured at the Wallace Weir facility in the Yellow Bypass. So this is actually funded by DWR through Region 2. So as adult Chinook salmon returning adults get sort of trapped in the Yellow Bypass, they will eventually hopefully show up at the Wallace Weir trapping facility to where adults are trapped. If they're still, tissue samples are taken from all of them. If they're still alive, they're actually then transported over to the Sacramento River and released. So over the last 2 years, we've found 500 individual adults were fall/late fall Chinook. We found 41 spring Chinook and 55 winter Chinook adults, which is a little concerning because winter Chinook are one where their populations is so low. I mean, they're fully protected ecotype of Chinook salmon. So they're one where every single reproducing adult matters. So our deliverable on that are just periodic reports to Region 2, where we list out the samples that were collected and which stock they identified to. So just kind of another example of current projects is where we are doing monitoring of hybridization in Independence Lake. So Independence Lake is a high elevation lake up in the Truckee Basin, or Truckee River Basin. It was historically a stronghold for pure, genetically pure Lahontan cutthroat trout. LCT are federally listed as a threatened species, and there's a very active kind of management, conservation, recovery group with a lot of different agency and NGO partners. In 2016, it was discovered through another project, one we weren't doing, but a UC Davis project, that so rainbow trout seem to have gone into Independence Lake and hybridized with our pure stronghold of LCT. So the thing is with hybrids, once they're formed, now kind of the cat's out of the bag, so to speak. So what we've been doing is we're trying to monitor and get an idea just for how serious the problem is. How many hybrids are we seeing? So they've recently started trapping fish in the lake that are trying to ascend the main water source for Independence Lake to go up and spawn. So we're trying to physically kind of block them from spawning, individually pit tag them, take a tissue sample, so that we can genetically test them and determine, is this actually a pure LCT or is it one that's a hybrid? So that when that fish shows up again, they know which ones they can spawn and who should be allowed to spawn and who shouldn't. I mean, it's a lot more complex than that, but again, this is one that we're kind of actively involved in. Okay, so another current project, been kind of our big lift in the current fiscal year, is looking at genetic diversity of trout in the Kern Basin. We have SWIG funds right now for a study on California golden trout. We've also been examining genetics of little Kern golden trout in parallel. Kristen's been working on that side of it, as well as a bunch of other populations from the Kern Basin. This is probably, aside from anadromous salmonids, this is probably the most intensively studied and surveyed fish genetics topics, in our state, going back probably at least 20 to 30 years or more. As everyone knows, the California golden trout is our California state fish. Little Kern golden trout is, and the little Kern golden trout is federally listed as threatened. Intergression with other strains of Omicus, mainly hatchery strains, has been a long-standing concern. Everybody knows we've had fish have been planted all over the state, for decades and decades. So we're kind of looking at the questions of, we have many different sort of, say, stronghold or possible stronghold populations of these fish. And we just want to know, okay, which ones are still pure? Have they become hybridized with other, say, out-of-basin strains of Micus? And also, too, as an aside, if we were at some point going to start, a hatchery-based program, in the Kern Basin to use a native stock from the Kern, what would we use? Where would we go? Which stocks would we look at? So this is something we're actively still chewing through the data kind of as we speak. I actually have structure running on my computer now. But again, our deliverables at the end of this are going to be genetics management recommendations, to the department. What's the best way to manage these different subspecies going forward to try to maintain their ability to adapt to future changes? Again, kind of that concept of establishing resiliency, looking at sort of how their patterns of genetic diversity are apportioned across the different population units, and which ones are pure, which ones may be hybridized in general. So here's kind of a newer PCA plot from some data we're currently working on with these. Again, I'm not probably like everybody that's used, probably everybody who's used RStudio knows that you might spend days asking Dr. Google how to get us to do one simple thing. But I don't know if, again, I'm not sure if people could see my mouse or not, but kind of our shotgun blast of data points in the top right that are in yellow is our little Kern golden trout from the project Kristen's been working on. The ones in green are other populations from the Kern River. Ones on the left in black is actually California golden trout, our California state fish. Then down in the lower right is probably at least 20 different sample sets of fish from outside the Kern Basin. So again, just trying to see within the Kern how our populations, where our population boundaries, which ones are more or less diverse, who might be hybridized with who. Again, just trying to inform what's the better way to manage these fish going forward. So this one plot here has about 3,000 fish on it. Again, 96, started with 96 markers, and then altogether on here, there's probably about 100 different individual actual sample sets. Okay, so we also run what Kevin Schaefer likes to call the genetics help desk. So we are often asked to provide basically technical expertise on different management questions and recovery questions on a variety of things besides salmonids. So for instance, Sacramento perch is kind of our only native centrarchid in our state. UC Davis has done quite a bit of genetics work on that species. So we have a pretty good database to go off now of how the different population units are related to each other. So we're kind of using that to try to develop a plan, through Fish Branch, going with the regions about what's the best way to manage Sacramento perch going forward. Also involved with the tui chub and arroyo chub. So tui chub are, I believe, fully protected. Arroyo chubs are a native chub species to Southern California that are not listed. I think they're a species of special concern. Also involved with Santa Ana sucker translocation questions, and they are a federally threatened species. And then also unarmored three-spined sticklebacks are fully protected. Again, a native, not salmonid in Southern California. So I'm going to talk briefly just about some future projects. So these are all externally funded in the sense that we've had to work with regional staff and branch staff to develop project ideas and align out project scope and deliverables, and then apply for and compete for SWIG and Section 6 dollars to do these projects. So these are ones, three that we've written applications for that we, fingers crossed, are going to be funded here hopefully pretty soon. One is, the first one is to look at genetics and genomics of interior red bands. Kind of the upper Pitt River watershed, Modoc Plateau is kind of the last sort of really unexplored area in the state in terms of genetic diversity of native trout. Myself and Kristen have been working and framing up a new project on developing some eDNA assays for native fishes. And I'm sure she's going to talk about this in a bit in hers, so I won't steal her thunder. And then the bottom one is a genetics/genomic study on Lahontan cutthroat trout to basically do a fresh statewide evaluation of all of our LCT populations. Again, they're federally threatened. We've been through some periods of extreme drought. We've done some translocations, and we've had issues, as saw in a few slides ago, with hybridization of LCT and Independence Lake. So there's tons of questions there. So kind of some new projects in the pipeline. I'm just going to talk very, very briefly, because people have heard about this a bit, as the California Conservation Genomics Project. I'm going to keep going to wrap this up so I can hand this over to Kristen. So this is a UCLA-led effort to sequence 150 individuals from 100 species statewide. Generally, they're just trying to determine on a meta-analysis level if there are hot spots for genetic diversity across multiple taxa. It's funded by $10 million from California State Legislature. Our laboratory, we're actively collaborating on projects for rainbow cutthroat trout, as well as tui and arroyo chub. The LED staff, forensics and genetics lab are collaborating with some other researchers on black bear and directly on mountain lion. So how do you request our services? So we can do interagency contracts. We are a state laboratory. We might have the capacity to do the project. So if you're maybe on the line from DWR or another state agency, we might be able to help you. So if you come to us, generally a project needs to be self-contained with funding for cover supplies and expendables, as well as lab tech time. We'll certainly work with you to develop a budget for your proposed project, as well as sort of fleshing out, well, what are the questions you're trying to answer? Is this something we can truly help you with? And then on our end, what's really the most important and appropriate tool for us to use to help answer the questions at hand. As I mentioned, projects need to be generally financially self-contained, whether they're reimbursable, Section 6 or SWIG. Generally, we like to set projects up, to meet this what we call the SMART objectives. So folks in our department that have been involved in grant proposals, writing, review, are certainly familiar with what those are. Again, we typically, we work extensively with both branch and regional staff to help them develop some project ideas, funding sources, and see, what can we do to help. I mean, we are an agency laboratory, so it's really our prerogative, to try to help, as many folks as we can, within the constraints of our current staffing levels. So it takes a village to keep a program like this, get the program like this up and going and running. So our Fish Branch admin staff has been indispensable in helping us get this get the genetics lab and program up and running. We work extensively with our Central Valley Tissue Archive, as well as with our facilities folks, and then also Tony Warrington with LED has been hugely helpful in sort of coordinating, sort of the three facets of our genetics lab and helping align funding and coordination as well to kind of keep the laboratory as an entity, get it up and running and keep it going. So there are other folks that do work out of our genetics lab. Law enforcement division, again, Tony Warrington's sort of their main point person. In wildlife branch, Dr. Mike Bukowski runs a very similar program than we do on the fishery side. His program is focused on wildlife. So folks you want to reach out to if you're interested in, certainly reach out to those folks if you're interested. So I'm going to go ahead and try to hand this over to Kristen. Again, this is my first time kind of doing this through Skype, so I'm going to go ahead and hand this off to Kristen. Again, we'll hold questions till the end. Let me find the stop sharing question, and we'll get Kristen queued up here. Yeah, so just bear with us for a couple minutes, and we'll get Kristen up and running here shortly.
Okay, thank you very much. Well, I'm just going to jump right in then, and probably the best place to start would be with what is environmental DNA. So this is just DNA shed from an organism into its environment. This could be through feces, shed skin cells. This could even be coming from a carcass. So what we would normally sample to obtain this environmental DNA is either water, we could use sediment or soil, or even air is an option, believe it or not. Probably the most researched and most common is water, but don't make this out to be that this can't be for terrestrial species as well. We typically target mitochondrial DNA instead of nuclear DNA for these studies, just because there are so many more copies of the mitochondrial genome within each cell, and we're usually dealing with pretty degraded and trace amounts of DNA, so really low concentrations, and mitochondrial gives us a better chance of finding a species we're looking for. So this is a rapidly growing field, and I'm going to kind of talk about some of the applications that might be relevant to you. We can start with more of a basic presence-absence test for a species. I use absence a bit loosely. I'll come back to that, but we could use this for invasive species early detection. In the case of our first in-house project, we were using this to determine the eradication success of non-native trout before the native species was reintroduced. This can be really helpful for rare or elusive species that may be hard to detect using more typical field methods. We could use another approach where we get a full community assemblage of maybe a group you're interested in looking at, and we could compare diversity across sampling sites. This could be used really broadly as a range-wide study of species. And there is also a possibility of using these methodology for looking at relative abundance. This is kind of in the early stages of research, so I'm not going to go into that in too much detail, but I imagine there's going to be a lot more refinement in that area coming soon. So what do field methods in eDNA look like? If you were collecting soil or sediment, you might just use a soil core sampler or net to collect sediment. For filtering water, we usually use 1 to 10 liters. Our protocol uses 5 liters of water, and we use a special filter that's going to trap the DNA on the filter. That's what the top left image is showing. So that contains the DNA. That's what we extract the DNA from. There are a lot of different types of filters with different pore sizes that may have better efficacy. And there's the option to filter this in the field, which is what we recommend, versus collecting a water sample and then having us filter it in the lab. Takeaway here is that there are many nuances to this methodology, beginning with the very first step. There are some other field sampling possibilities as well. More recently, there have been some studies published, especially rare carnivore studies looking at tracks, either through snow or soil, and being able to actually ID the species from the track using this eDNA method. Urine in snow is certainly an option as well. Our wildlife forensics lab actually used this technique in a case of mountain lion poaching where they were able to identify the suspect's dog at a remote site where the carcass was found. So there are some really creative options to using this technology. And the middle image is an eDNA sampling backpack that is made by Smith Root, and that just kind of speeds up the filtration of samples, which can actually take quite some time, especially if there's a lot of turbidity in the environment you're sampling. So there's all these fancy field sampling tools out there now as well. So when we're thinking about eDNA sampling design, one of the first things we kind of have to think about is how far can eDNA travel if we're talking about a freshwater ecosystem. And unfortunately, the factors affecting the way eDNA disperses in water varies greatly and varies significantly by system. So ideally, you would want to do a pilot study in your system to get a better idea of this. But there is research out in the literature where you could kind of pull pieces of information and get an idea of what you might expect to create a more informed sampling design. So from where the species occurs to detection downstream, researchers have found in fish and small streams, we might be able to detect that fish 360 meters downstream at most. There's a much wider range in invertebrates. We might be able to detect invertebrates 1 kilometer up to 12 kilometers downstream in rivers. So you can imagine that could complicate your results. If we're talking 12 kilometers downstream, that doesn't necessarily mean the invertebrate, maybe invasive species you're interested is right there at that sampling site. And then we also need to think about how quickly eDNA degrades in the system. If you are interested in a migratory species, for example, or a species that you want to see if you eradicated successfully, and there could be carcasses in the environment still. In water, eDNA could persist for days up to weeks. Sorry, don't have very specific time frame there, but the literature is really wide ranging. There are a lot of abiotic and biotic factors that affect how long eDNA can remain in the system. So more acidic conditions break down eDNA more quickly, UV exposure, microbial activity. And then if we're looking at soil or sediment, eDNA seems to persist for much longer in these cases, for months, and even in some cases, studies have found years later they were able to detect a species that hadn't been present. And some important considerations. The efficacy of these methods can vary significantly. Just within the same site that you may be sampling, be interested in sampling over, say, several months or repeated years. In a drought year versus a wet year, we might have a very different likelihood of detecting eDNA, high flow versus low flow conditions. If it's at a time where there's a lot of agricultural runoff, some of the contaminants in runoff can degrade DNA really quickly. So as you're gathering a lot of nuances to what's going on here and how you might be interpreting the data. And the best way to interpret this type of data would be to look at it alongside the ecology of your target species, alongside any field data you have, not in a vacuum, and definitely be considering the ecology of your study area as well. So this is not a replacement for field work, but it can be a really powerful complement. And it's just like any other tool you may commonly use, it does have its limitations, of course. Another set of confounding factors that we need to consider is, again, if you're doing some sort of eradication effort, carcasses can provide a source of eDNA for perhaps up to a month, probably longer in the sediment. And there's always going to be this issue of transport of eDNA from outside your study area. So that could be you collecting the sample. Proper decontamination of your gear is vital just because, again, we're detecting such trace DNA using very sensitive tests. And you really can't control if boats are moving through the system, predators and scavengers. A really interesting study looked at how long after fish predation eDNA could be found from bird feces. And it was about a month later, they were still able to get a hit for that species. So if we have, for example, a 5-liter water sample, we've filtered that water sample, we've extracted DNA from that filter. In our lab, we are definitely planning to archive all of our eDNA samples because we have a snapshot of biodiversity in that system at that time. And while in our case, in our most recent study, we were looking for non-native trout, we know there is a lot of other DNA present in our sample, and it's possible that these could answer future research questions as well. So this is potentially really valuable. So there are two main approaches that we could utilize if we're interested in doing an eDNA study, and they're going to depend on what kind of information you're seeking. So the kind of more basic, cheaper, faster way is if you have just a target species or maybe a group of target species you're interested in finding out if they're present or absent, so to speak, this is probably the best approach for you. There are a lot of primers that are already available and published, and I'll provide some sources for you to research that to see if your target species of interest protocols have already been developed for identifying them. But this is going to answer probably more of a monitoring or a management question, something a bit more targeted. Versus a metabarcoding approach is where we would sequence all of the species in an eDNA sample or a group of interest, so maybe all of the fish species in that sample. So as you can imagine, if we're sequencing everything, then that's going to be a lot more costly. That's going to take a lot more time. Not so good if this is like a time-sensitive monitoring or management project. But you can answer a lot broader ecological questions, and you can compare diversity across sampling sites, for example, and look at just a very different set of questions. So if we wanted to go down the road of target species ID, these two gray shaded bullet points apply to both of the methods I just explained. Decontamination, both in the field and in the lab, is of critical importance. So we have a dedicated space where we do this work, and none of our other population genetic studies occur within this space because of this trace DNA that we're working with and this really sensitive test that we're using. So then when we would do DNA extraction from these filters or from a soil sample, and in target species ID, we would use quantitative PCR and do this in a series of replicate reactions. And basically, in these replicates, did we get positive hits or did we not? And that's the end of our analysis. Whereas if we're talking about metabarcoding, what we're going to be using is some universal primers that maybe are just targeting all the aquatic invertebrates in your samples and not species specific at all. And then we're going to sequence all of those reads. And the breakdown here can be in that we have these sequences that we've obtained, but now we have to compare them to a reference database. This is where other researchers are uploading sequences and saying, hey, here's the sequence for these given species. We know it's these species. So if in your study area there's a high level of endemism, we might not be able to find them in the reference database or identify them in any way. It's rapidly expanding. Researchers all over the world are uploading to these databases, but there can still be some problems. Your study is limited by the reference database. So my opinion is that eDNA plus field methods are often more powerful than either alone. and can be a time-saving method. eDNA could be a non-invasive first step where you don't have to get your hands on a species. And it can be a way for you to target your field sampling or inform your field sampling design. So is the species within the area, and do you want to go in and do a more intensive, say, electrofishing survey where you're going to get age class information, measurements, things, information you can't get from an eDNA sample. Likewise, if you wanted to do a population genetics study, like Josh explained, we can't do that kind of work with eDNA because we don't know how many individuals are within our eDNA sample. So we need actual tissue samples. So again, eDNA could be the first step for you to figure out your sampling design to obtain those tissue samples from individuals. And I mentioned the target species approach to be often called a presence-absence test. I somewhat discourage this title just because, as with any field technique, we can never prove absence. eDNA is the same. So we would say something like, since it's unlikely, given hopefully field data that we present alongside the eDNA data. And I know there's a lot of questions usually about invasive species. And if we get one positive sample across, say, hundreds of samples in a study area, that one positive sample has really high stakes, may even result in some amount of panic. But I would say if you're going this approach, there needs to be some pretty strict protocols in place for how to deal with that positive sample because contamination issues can be high. And this method's not perfect, of course. So this should just trigger further eDNA sampling within that area, surrounding where the positive hit was. We need to rule out contamination and then probably trigger a larger field survey as well. I was just going to briefly touch on this range-wide study that the U.S. Forest Service Lab is conducting on bull trout. This is kind of like the grand picture of what eDNA could accomplish. So they're looking at partnering with 20 different organizations to basically go out and collect samples and determine if bull trout is present or absent. Absent, you can go online and interact with this website and look at different regions. But for species where we don't have a lot of information about them and where they occur, something like this could potentially be beneficial, and we may not be there yet. Our lab could probably take on a smaller study currently, but eventually we hope to create more lab space where we can do larger scale studies. We're kind of in the process of acquiring a larger dedicated eDNA space to take on potentially more projects or larger projects, but no definite date on that yet. Of course, these things require time, staff, money, but my opinion is that in the long run, we end up saving time and money and staff if we don't have to maybe do as much field work to accomplish the same goals. So kind of in summary, what is the best use of eDNA? When conventional survey methods could be difficult logistically, so if you have to get a large crew out to a remote location, a lot less people may need to be involved with eDNA, and there may be a lot less equipment. And given our current environment, I know a lot of you have had to cancel field seasons or things are up in the air, and sometimes eDNA sampling can be done with just one person and definitely can be done while social distancing. So given the state of affairs currently, this could be a good substitute for some goals of what was previously a larger scale field survey. And of course, if there's take associated with listed species, eDNA is a great non-invasive method. And if you have issues detecting a rare species in the field, a lot of times eDNA actually has a higher detection rate than common field methods and is less costly. So kind of in closing there, try to think about how eDNA could complement rather than replace your field surveys. Just a little list of some of the published eDNA assays, not just aquatic organisms, have terrestrial mammals, as well as a lot of wildlife diseases actually being tracked and monitored through eDNA. So I'll leave you with, if you want to take a quick picture, to have some resources where you could find some more information. The third bullet point will give you some information of where you can look for published assays for a target species you might be interested in. I'm happy to provide any references for any of the information I've mentioned if you want to email me. And then that final bullet point, there's some really great talks available for free from leaders in the eDNA field, a conference that was held at UC Davis earlier this year that I think will be very valuable. And with that, we'll leave some time for question and answer.
All right. Well, thank you both so much. That was really great information and very well presented. I'll admit I did not know a lot about this topic coming into today, and I just didn't realize how many opportunities there were to actually apply this information, this data, to actual management decisions, different kind of long-term, short-term conservation questions. So this has really helped kind of ground this field, this science in reality for me. So very much appreciate the presentation today. I'm going to go ahead and turn to the questions here. We've got several in the queue for you guys. Let me scroll up to the top. And if Jeff and Kristen, if you're good with it, I'll just kind of start to go down the list.
Yep, sounds good.
Sounds great.
All right. Well, the first question came in, Jeff, the first few came in during your presentation, and this one in particular came in on your slide when you were talking about and kind of defining effective population size.
Okay.
And the question is, which N or NE is used most frequently in fisheries? Inbreeding, coalescent, or variant NE?
I would say it depends. If you are, so it kind of depends on the program. To calculate a variance NE is you actually need information from the actual hatchery breeding program, in the sense where you have, like, I know how many fish I spawned, I know how many males I used, how many females I used, and then I also know, how many, what my different family sizes were that I made. So, for instance, it's used routinely in kind of our winter in the evaluating the winter run Chinook program, in their yearly breeding. As far as estimating any, a lot of times you basically just have genetic data, like from a survey of a wild population where you know little to nothing about how many fish, how many families you have and that type of thing. So you're estimating more, say, from like linkage disequilibrium or changes in heterozygosity over time. There's different ways to estimate it. And again, it kind of depends on the situation. Like I said, if it's more of a hatchery one where you have really good records, you can do a variance NE. If you're just looking at a natural population, there's other ways to estimate it. So I hope that helps.
Yeah. All right. Oh. This next question came in, Jeff, when you were talking about some of the costs associated with equipment and some of the work that you do. Mm-hmm. And the question is, how does internal lab costs compare to outsourced, I'm going to say Tackman style SNP markers? And is 96 markers enough to capture LD in the population under study? And I apologize, I don't remember what LD stands for.
Yeah, linkage disequilibrium. Again, it kind of...
Yeah, that one.
Yeah, I mean, again, it kind of depends. It's, if you have a population that's pretty highly variable, to where you have, actual, both alleles at a SNP, in those 96 different markers, you should be able to make a reasonable estimate of it. If you're looking at a population that's genetically depauperate, to where you have, meaning that there's very little genetic variation, and so you have 96 markers, but there's only one allele of each, then you then your estimates get a little wonkier, so to speak. That's a statistical term. That said, even if you have, say, like good microsatellite data and stuff, you can run it through like some of the different packages out there for estimating effective size. And even though you get a point estimate, sometimes it's not uncommon to have your confidence intervals be essentially positive to negative infinity. So yeah, estimating effective size directly from marker data is you do get a point estimate. It really depends on how variable your underlying population is. One thing I didn't mention is for some of the trout work that we're doing now, is obviously a lot of it's been done using the 96 SNPs. We are actively working on incorporating or building up our data set using what's called microhaplotyping. So those are, I think, a set of 120 additional markers that we genotype directly by sequencing our PCR products on our Illumina sequencer to add, 120 more markers to our baseline data sets, primarily for that purpose and a couple other ones. So yeah, I guess the answer is the accuracy of estimating that from SNPs depends on how variable those SNPs are in your population.
Right, next question. Why is hybrid vigor bad? Is it an aesthetic thing? And then a comment, aesthetic is fine as a trait to select for from an angler/sport standpoint.
Well, that's about 5 different questions in one, but I'll do my best to answer it. So this question of hybrid vigor. So hybrid vigor so I think I'll answer that by saying it depends on what the purpose of the fish is. So if you're producing like hatchery trout for the purpose of recreational angling, then sure, you probably want a nice big vigorous fish that fights well. And has a really good return to creel, good survivorship in the hatchery, disease resistance, and in that sense, a lot of times that you want variability in those fish. You don't want things that are inbred and just don't perform well. If you're looking at stuff, so that's looking at it through a recreational angling lens, which is obviously a big part of what our department does. If you're looking at things through a conservation recovery lens, a lot of the conservation and recovery objectives, or however you want to phrase it, are related to either a species of conservation need or one that's actually listed. A lot of times a listing is based on identifying kind of unique genetic units, say like LCT or say like little current golden trout or something like that, to where the unit that's listed is kind of a unique genetic lineage that has like has trying to think of the right way to explain ESU and something that makes sense. So essentially, you want to preserve these sort of individual components that are, as we call, important to the evolutionary legacy of the species. So if you have all these different units, again, speaking of the portfolio effect, that are now all blended together, yeah, maybe they are. You start losing some of those individual lineages that you're actually trying to protect over time, because each of those lineages may be, well adapted to whatever they are currently living in. Say like the Upper McCloud, or say like a high elevation stream, versus like the Central Valley, for instance. So the answer, it really depends kind of at what lens you're looking at that question through. Again, because we try to do a lot of different things to a lot of different stakeholders, and our approach to each is very different, depending really what the purpose of those fish is.
Right, I'm going to keep rolling through these. The next question came in, Jeff, when you were talking about some of the future genetics projects and types of things that you're going to be working on, and the question was just fish. Can we get help with other listed plants and animals if we can get the funding? So I think the question of, are there other types of projects that could be done?
Well, I think your dog agrees, Whitney. I hear him him or her in the background. So I mean, myself, I work for a fisheries branch, so I'm directly responsible for a fisheries genetics program. So as I mentioned, the genetics lab is also home to Dr. Mike Buchalski in the wildlife genetics program. So he has a role very similar to mine. So for wildlife-specific questions, he'd be the one to talk to. We don't in our lab. We don't have anybody right now that is doing really doing plants as a plant geneticist, or marine species for that sense. I mean, our lab's pretty small just in terms of physical constraints. So those would be other areas we'd probably like to go in the future. But and then as our lab is also shared with folks from our forensics, from LED's forensics lab, which is physically next door. So there's, right now there's 4 full-time people there, and they're working on various genetics projects as well. I know I think one of their folks is working on a Dudleya project, because Dudleya are a succulent that I think is illegally harvested and trafficked in California. They're also actively involved in determining, tracking all of the new wolves that are coming into California, trying to estimate which ones are actually, quote, a pure wolf versus something that's a hybrid with a domestic dog. So they're involved in a lot of other stuff as well. I mentioned, Tony Warrington's a retired assistant chief, and he sort of manages that group of folks. So yeah, but as far as plants and marine, we don't have anybody right now. We just don't.
And that question came from Kelly. If you needed to or were wanting to follow up with any of those folks, feel free to email me, Whitney Albright, or Jeff, and maybe we can help make those connections or give you those names again.
Sure.
All right, next question came in, Kristen, during your presentation. The question is, how feasible is it to enhance a presence/absence eDNA study with genomics and genetics? The context here is that we want to identify tributaries that have rainbow trout, but may also want to know how connected these populations are.
Yeah, so I think the best approach for that is maybe to do your eDNA presence-absence study first, see where you are finding rainbow trout occurring, and then unfortunately the best approach would be to go in and do some electrofishing or get your hands on some actual fish and collect tissue samples so that we could do a bit more population genetics work or find out the level of genetic connectivity between these different groups.
All right, next question. Has anyone looked at the possibility of using eDNA in the marine environment, or is that way too open of a system?
Yes, definitely. There's actually a ton of research published on the marine environment. I don't have a ton of information on that other than I know it ranges from invertebrates up to marine mammals. And you could do a quick literature search and find a lot of information. If there's a specific question you're interested in, I have a feeling you will probably find some hits on it.
Yeah, I'll follow up on that too. We've been involved, sort of directly or indirectly with some eDNA projects looking at detection of delta smelt, in the the San Francisco estuary, and doing eDNA work in an estuary environment where water basically is either stagnant or just sloshes back and forth is a very, very different, say, model or approach, and the challenges are very different than doing eDNA, like in a stream where water's fairly clean and flowing in one direction. And also, too, if you're doing eDNA collection in an area with high turbidity, can be a lot more challenging, is because your filters clog easier. So yeah, there's tons of applications out there for eDNA. Of course, they all have their own unique challenges. So if you're interested in sort of like an estuarian species, whoever asked that question, if you're still on, there's I want to look up a study that Andrew Kinziger published maybe a year or two ago on an eDNA project with Tidewater Goby. They kind of give you an example too of some work that's been done.
All right, and let's see, next question. What is the collecting method for terrestrial species, or is that even possible?
It is possible. If we're talking about eDNA, I presume. still.
Yeah, I think this one might have come in right at the end of yours, Kristen. So I think it's
Okay.
I think it's directed at eDNA.
Yeah, definitely going to depend on your species, but soil is an option if you're able to identify tracks. There have been some studies recently that whether soil or snow, that can be an option if that's a challenge. I would say soil, or potentially even things like swabbing environment where you expect the species to occur could be a possibility. And you would be surprised also by the terrestrial species you might be able to identify in an aquatic environment as well, especially if there aren't a lot of water sources nearby that they might be utilizing a single water source or congregating in that area from time to time. Then you can actually, even if they're not living there most of their life there, you might be able to identify them at those aquatic locations.
All right. Well, I want to take a minute and check in here because I realize we're right at noon. We do have just a handful of other questions. Kristen and Jeff, are you available to stay on the line for a few more minutes, or do we need to go ahead and wrap up?
I can stick around for a few minutes. Let me just check my calendar real quick. I think I'm okay here for a little while. Hang on one second, Whitney.
Okay.
Yeah, I'm
I'm
okay.
available and happy to answer further questions.
Okay. Yeah, I'm free till 1 o'clock.
Okay, great. Well, we just have a few more here that I want to try and get to, and but for folks on the phone, if you need to drop off, completely understand, but we'll hang on here and see if we can get through the rest of the list. And the rest of these questions, I think, started coming in kind of after both presentations were complete, so they're kind of up for grabs. The next question and comment is, I read a study where flowers were sampled using metabarcoding to get insect species using the site. Could be promising for the bumblebee species that we're potentially listing. Have we conducted many terrestrial sites? It kind of ties to a couple of the other questions, maybe.
Our lab has not done any terrestrial sites. We have only completed one eDNA project and have one in the works. But yes, I've certainly seen a lot of references to such studies in the literature, and I think there's a lot of potential for branching out into terrestrial environments. It sounds like in this case they might be doing something like swabbing the flowers or something, which is interesting, and kind of like we're going in all these different directions with eDNA and the possibilities.
Yeah.
So that's really interesting.
Yeah, speaking to the metabarcoding question directly, there's there's a lot of work, as Kristen alluded to in her presentation, there's a lot of work going on, in the scientific community about how to properly apply, metabarcoding approaches to eDNA. Let's say metabarcoding was something that kind of first came out, I believe, like through basically doing gut analysis and trying to characterize microbiome communities. But as far as applying it in an eDNA context, it's a bit of the Wild West right now, to be honest. But I mean, we actually do, in our laboratory, we have a, MySeq sequencer, to do some limited, next generation sequencing work. We had in fact, one of the full-time staff members in the forensics lab next door did a metabarcoding project in our lab. She was looking at microbial communities of black bears as a function of whether they're like a captive bear or an urban urban bear versus like a wildlands bear. So it's something we've kind of done a little bit of in our lab, but like Kristen said, doing true like metabarcoding on a for like a eDNA type thing is still a bit of the Wild West, and those are very complicated projects to do, much more so than it appears on their face. But it is a direction the field's moving towards.
Okay, this next question is for Jeff. Esther asks, how many samples do you recommend obtaining when trying to determine effective population size for a terrestrial carnivore, as an example?
Oh gosh, For a terrestrial
Yes.
carnivore? Yeah,
Oh,
it's yeah, no, and that's totally fine. I mean, I've done quite a bit of wildlife work. You know, when I worked for LED, you know, on big game. You know, generally, you know, for estimating effective size is I don't want to say it's a pretty old parameter, but it's not something that's new and people have been doing for you know, for quite a long time in a variety of markers. I generally recommend when people are collecting samples is do probably somewhere in the range of 30 to 50 individuals per population if you want to estimate metrics from it. And that kind of goes for a wide variety of the population genetic metrics, you know, effective size being just one. I usually set, you know, 30 is sort of the bare minimum. Me, I generally think more is better when you can get them, but I'm also mostly a fish person now to where, you know, we can go get a few hundred samples, you know, fairly readily in many cases. But I usually try to set probably, you know, 30, anywhere 30 to 50 is sort of the bare minimum.
All right, last question here. The question is, I feel like local adaptation and the desire to preserve that gets assumed too readily for listed plant species that have been severely fragmented. Is there an easy test to differentiate between local adaptation and inbreeding depression?
Well, that's a really good question. I will say I'm not a plant person, so the way I respond to that is more a function of my own background. We wrestle with that same question a lot of times in fisheries management when you have these very small, isolated, highly fragmented, like headwater populations, as we call them. You know, ones where you have really small populations of fish that have been like trapped above, you know, barriers, while everything below the barrier is either, you know, hybridized with hatchery fish or something else. So it's certainly an interesting question, and sometimes more a philosophical one than a scientific one. But what we try to, one way that we look at it is that you might have a population or a species that's highly fragmented. You have small groups now where they're maybe don't have a lot of genetic diversity. But when we look at it with fish ones, maybe they're really small fragmented populations. But if they've been persisting a long time, and not showing more of like the textbook signs of inbreeding depression, such as reproductive failure, you know, deformities, things like that, then probably tend to leave them alone in many cases. It's always, yeah, it's a tricky question to get at. And said we struggle with that same thing in fisheries management. But again, like we said, we try to, you know, and that's kind of like when you do a population genetics study and you get, say, estimates of effective size, or you're looking at these and they say have a handful of populations really have no diversity left. That's probably the result of selective sweeps going through there, as in addition to effects of genetic drift, removing variation just by chance alone, as opposed to an adaptive process. So in a lot of these natural populations, you have competing processes going on. You have adaptation driving genetic diversity in one direction, and then random processes like genetic drift and negative effects of inbreeding driving it in the opposite direction. So it's, like we say, a lot of times with small populations, you're kind of driving with one foot on the gas and one on the brake at the same time. So it's tricky because you have a lot of competing forces. But like I said, when it comes to like fisheries questions, you know, if you're looking at a population now where you just know they're just not doing well, like not reproducing well, you see deformities, things like that, that's probably a pretty good indication that you have inbreeding depression. You know, on the large mammal side, you know, I think, you know, Thule elk are probably a pretty good example of that. You know, many years ago, I was involved in a Thule elk or statewide elk genetics project. I mean, it's remarkable how much less diverse Thule elk are. They think they could have gotten down to a single breeding pair at one time, and you know, they have certainly their fair share of physiological issues. You know, probably likely as a result of inbreeding depression. So anyway, it's yeah, it's a good question, tricky one to answer, but anyway, I hope I gave you some different perspectives on it.
All right. Well, that's all we have in the way of questions from our participants. Any final comments, thoughts from either of you, Jeff or Kristen?
Yeah, I would just like to thank everybody for joining us. I know we still actually have like 60 people online, watching, listening to questions and things. So I certainly want to thank everybody, for was able to take the time out of their day and register and join in. And again, thank you very much, Whitney and Science Institute for running the series and accommodating us and helping us make sure we're got our presentations up nice and seamless. So thanks again, everybody. And if you have questions, certainly feel free to reach out, to myself or Kristen as well.
Yeah, I second that. Thank you, and feel free to shoot me an email if there's any questions that come up later.
All right, with that, we'll go ahead and close out. Thanks everyone again for joining. As we mentioned at the start, we have recorded this and we'll be posting it to the kind of archive page on the website. So if you know of other folks that might be interested and missed the talk today, feel free to point them there. And yeah, appreciate you all supporting the lecture series, and please join us on July 30th for our talk next month. And yeah, we'll see you then. Have a great day, everyone.
Okay, bye-bye, y'all.
Thanks.